Hello, this is the Unit 3, Module 10, Chapter 20A presentation. The big picture for this Part A presentation includes a bit of discussion about the history and development of antimicrobial drugs, the features and characteristics of antimicrobials, and the mechanism of action for antibacterial medications that inhibit the synthesis of the bacterial cell wall. Keep the following ideas in mind as we progress through Chapter 20. The ne plus ultra of chemotherapy is to administer a drug to an infected person that destroys the infective agent without harming the host's cells. Keep in mind that antimicrobial drugs are produced naturally or synthetically, and consider that microbial virulence factors are targets for chemotherapy and drug development. Paul Ehrlich discovered in 1909 that compound 606, arsphenamine, effectively combated the Spirillum spirochete bacterial type, one of whose subspecies causes the sexually transmitted disease syphilis. Compound 606 proved to have few side effects in human trials, and the spirochetes completely disappeared in 7 syphilis patients after this treatment. Salvarsan, the trade name for compound 606, was the first effective medicinal treatment for syphilis, thereby initiating and also naming the concept of chemotherapy. It is an unimaginable world that has no usable antibiotics. Antibiotics have saved countless lives from common infections and disease. Even the evolution and misuse of antimicrobials are leading to new research in the fight against microbial disease. As early as 8,000 BC, the Egyptians used sulfur dioxide by burning sulfur to sanitize fermentation and storage equipment for wine production. The Egyptians also used plant extracts in the form of spices and oils as antimicrobials for the preservation of food, as well as for embalming. Before the early 20th century, treatments for infections were based primarily on medicinal folklore. Mixtures with antimicrobial properties that were used in treatments of infections were described over 2,000 years ago. Even the prehistoric peoples used a number of plants in wound treatment, and it seems possible that many plants have the properties of antimicrobial effects. Prontosil rubrum, whose commercial name is sulfanilamide, was a red dye found to inhibit some gram-positive bacterial species. Prontosil became the first commercially available synthetic antimicrobial antibiotic. Gerhard Domack, its discoverer, saved his 6-year-old daughter Hildegard from an amputation of her arm. She had contracted a severe streptococcal infection from an unsterilized needle. Both salvarsan and sulfanilamide are chemotherapeutic agents. Alexander Fleming identified that a mold he accidentally found growing in a petri dish upon his return from a holiday had antibiotic properties. Penicillin was originally described as an excreted fungal compound that was toxic to the gram-positive pathogen Staphylococcus aureus. This is Howard Florey, an Australian. He is responsible for the purification and mass use of the penicillin that Fleming discovered. The photographic panel in the slide shows just how awful untreated bacterial infections are. In this case, this was blood poisoning. The pictures of this child were taken in 1942, when the threat of dying from a bacterial infection was very real. Image numbers 1 and 2 show a young girl who appears to be very sick indeed, yet just a few days later, when photos 3 and 4 were taken, she seems much better. In the final 2 photos, she is completely cured. This amazing recovery was possible because Howard Florey and Ernest Chain worked out how to purify penicillin for clinical testing. Fleming, Florey, and Chain were awarded the Nobel Prize in 1945. So, how many antimicrobial drugs are there? Well, there are well over 200 different types, basically classified into 20 drug families. Most of them are antimicrobial drugs for treating bacterial infections. Selective toxicity means that a drug should harm the pathogen, but not the host. In treating a bacterial infection, it is best to choose a drug with the highest level of selectivity, but the lowest level of toxicity to the patient. The selectivity and toxicity are measured by the chemotherapeutic index, also known as the therapeutic index. The therapeutic index, or TI, is the ratio of the dose of the drug that is toxic to humans as compared to its minimum effective dose on the pathogen. A high therapeutic index number is desirable. The toxic dose of a drug, again, is the concentration causing harm to the host. The therapeutic dose is the concentration eliminating pathogens in the host. Now, the antimicrobial action of a drug may be to slow or stop growth. This is bacteriostatic. The action of the antimicrobial may cause death of the cell. This is bactericidal. So, a bactericidal drug kills bacteria, whereas a bacteriostatic drug inhibits their growth but does not kill them. Some examples are given in the figure. The key features of bacteriostatic drugs are that, number 1, the bacteria can grow again when the drug is withdrawn, and number 2, host defense mechanisms such as phagocytosis are required to kill the bacteria. Bactericidal drugs are particularly useful in certain infections, those that are immediately life-threatening and those in patients who are immunocompromised. The range of pathogens on which antimicrobials will work is called the antimicrobial spectrum. Broad-spectrum antimicrobials affect many taxonomic groups. Narrow-spectrum affect only a few pathogens. Which bacteria are affected? Well, broad-spectrum antibiotics are active against several types of microorganisms. For example, tetracyclines are active against many gram-negative rods, chlamydiae, mycoplasmas, and rickettsia. Narrow-spectrum antibiotics are active against one or very few types. For example, vancomycin is primarily used against certain gram-positive cocci, namely staphylococci and enterococci. A chart like the one in the slide is useful to determine treatment options, whether or not the pathogen has been identified. Drugs are often given in combination for several reasons. Number 1, to treat serious infections before the identity of the organism is known. Number 2, to achieve a synergistic inhibitory effect against certain organisms. And number 3, to prevent the emergence of resistant organisms. If the bacteria become resistant to one drug, the second drug in a combination will kill them, thus preventing the emergence of resistant strains. 2 drugs can interact in one of several ways. They are usually indifferent to each other, i.e., additive only. Sometimes there is a synergistic interaction in which the effect of two drugs together is significantly greater than the sum of the effects of the two drugs acting separately. Rarely, the effect of the two drugs together is antagonistic, in which the result is significantly lower activity than the sum of the activities of the two drugs alone. Some antimicrobials cause side or toxic effects. These can include drug-related fever, rashes, photosensitivity, which means a rash upon exposure to sunlight, and bone marrow suppression. The non-pathogenic resident bacteria in the body can be reduced by antibiotic use. This allows pathogenic bacteria to overgrow and cause disease. Some microbes are resistant to antimicrobials innately. Some microbes are able to acquire resistance to antimicrobials. The acquisition of drug resistance can occur through spontaneous mutations in critical chromosomal genes. Drug resistance acquisition can result from the acquisition of new genes or sets of genes via horizontal transfer from another species. And acquisition of drug resistance can originate from resistance factors carried on plasmids that are encoded with drug resistance, or from transposons. Let's start to discuss the drugs themselves. How exactly do drugs, for example, act on the bacteria? Well, there are usually 5 different ways in which they can act. They can act to inhibit cell wall synthesis. They can inhibit protein synthesis. They can cause inhibition of metabolic pathways. They can inhibit nucleic acid structure and function, or they can interfere with the cell membrane structure or function. Keep in mind the central dogma as you think about the antimicrobials. And Table 12.1 summarizes the characteristics of the major antibacterial drug classes in some detail. Let's begin with antimicrobial drugs that affect the bacterial cell wall. Active cells must constantly synthesize new peptidoglycan and transport it to the proper place in the cell envelope. Some drugs react with one or more of the enzymes required to complete this process. Drugs in this category are bactericidal antibiotics. Penicillins and cephalosporins act by inhibiting transpeptidases, the enzymes that catalyze the final cross-linking step in the synthesis of peptidoglycan. Penicillin's beta-lactam ring structure interferes with bacterial cell wall synthesis. Recall that peptidoglycan cell walls repeating disaccharide units are cross-linked with little short amino acid peptides. That's the linkage that's catalyzed by the enzyme transpeptidase. Penicillins work because first they enter through porin channels in the bacterial cell wall. Once penicillins are inside the cell, they bind to and competitively inhibit the transpeptidase enzyme. So, the transpeptidase enzyme is also known as a penicillin binding protein, or a PBP. Penicillin-treated cells die by rupture as a result of the influx of water into the high osmotic pressure interior of the bacterial cell. So that's why penicillin is bactericidal. Penicillin has remained the most widely used antibiotic. Penicillins have been chemically modified and multiple forms are now active against many gram-positive and even some gram-negative bacteria. Many bacteria, though, have developed resistance to the action of penicillins. Some bacteria hydrolyze the penicillin antibiotic using so-called beta-lactamase enzymes. These resistant types are found in common gram-negative rods like Escherichia and Klebsiella. Some of these beta-lactamase enzymes have minimal activity against cephalosporins. We'll discuss cephalosporins in a moment. Some point mutations in these beta-lactamase enzymes possess activity against all penicillins and cephalosporins. These are referred to as extended spectrum beta-lactamases or ESBLs. These beta-lactamase enzymes are carried on plasmids that can be transferred from one organism to another. At the same time, the effectiveness of penicillins against gram-negative rods has been increased by researchers making a series of chemical changes in the penicillin side chain structure. These changes have been made to keep pace with the development of resistance to penicillin by the bacteria, a microbial arms race. These changes mean that penicillin, which was originally active primarily against gram-positive cocci, is now usable against many more types of bacteria, provided they use a transpeptidase enzyme. There are several other beta-lactam antibiotics which inhibit cell wall synthesis. The cephalosporins are broader spectrum alternatives to penicillins. They have a different ring structure. They block the cross-linking of peptidoglycan. All cephalosporins have a modified beta-lactam ring. First-generation cephalosporins are active predominantly against gram-positive bacteria. Successive cephalosporin generations have increased activity against gram-negative bacteria. Carbapenems are broad-spectrum drugs which resist beta-lactamase enzymes, for example, the antibiotic imipenem. There are other antibiotics which can inhibit cell wall synthesis. Vancomycin is a glycopeptide that inhibits cell wall peptidoglycan synthesis by binding to the substrate of the transpeptidase enzyme. It is used for methicillin-resistant Staphylococcus aureus infections and people who are allergic to penicillin. Bacitracin is another antibiotic isolated from Bacillus subtilis and is the key ingredient in Neosporin ointment. It interferes with the delivery of peptidoglycan precursors to the cell wall. This slide contains questions to reflect upon and use the presented information.